Good morning, students. Our today's topic is the genetic material is DNA. The proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase. They worked with viruses that infect bacteria, and that is known as bacteriophage. The bacteriophage attaches to the bacteria and its genetic material enters into the bacterial cell. The bacterial cell treats the viral genetic material as its own and subsequently manufacture more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the virus that enters the bacteria. They grew some viruses on a medium that contains radioactive phosphorus and some others on the medium that contains radioactive sulfur. So some viruses they grow on radioactive phosphorus in the culture, and other viruses they grew in the culture medium containing radioactive sulfur. The viruses grow in the presence of radioactive phosphorus contained radioactive DNA because DNA contains phosphorus. Similarly, viruses grow on radioactive sulfur contains radioactive proteins because DNA does not contain sulfur. So, radioactive phages were allowed to attach to E. coli bacteria. Then the infection proceeded. The viral coats were removed from the bacteria by agitating them in a blender by mixing them. The virus particles were separated from the bacteria by spinning them in centrifuge. So the Hershey-Chase experiment we can study under three headings. First is infection, when the bacteriophage infect the bacteria. Second is blending, mixing of that culture so that it will separate the virus as well as the bacteria. Then under centrifugation, the virus particles will separate and bacterial particles will separate. So write the three headings: infection, blending and centrifugation. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria, because the virus will transfer its genetic material into the bacteria, so that the radioactive phosphorus which is present in the bacteriophage will go into the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive because sulfur is not the part of the DNA. This indicates that proteins did not enter the bacteria from the virus, because in the virus the protein coat is made up of proteins and genetic material. Only genetic material enters inside the bacteria and outside coat remain outside. So this indicates that proteins did not enter the bacteria from the virus. DNA is therefore the genetic material that is passed from virus to bacteria. This is an important experiment by Hershey and Chase. Next is, what are the properties of genetic material, that is whether DNA versus RNA. A molecule must have the following properties which has to act as a genetic material. First, it should be able to replicate. Second, it should chemically and structurally be stable. Third point, it should provide the scope for slow change. That changes should be slow, that it should be able to evolve. Fourth point, it should be able to express itself in the form of Mendelian characters, in the form of genes or characters. So these are the characters that must be fulfilled by a molecule that has to act as a genetic material. Both the nucleic acids, DNA and RNA, have the ability to replicate, but proteins fail to fulfill this criteria itself. So DNA and RNA can act as a genetic material, but protein can't. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. The genetic material should not change with the circumstances, outer circumstances. It should not change. Stability is one of the properties of genetic material. Was very evident in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the properties of genetic material. In Griffith's experiment, we have seen that heat killed DNA has some properties which can transform the R stain bacteria into S stain. So this now can be easily explained in the light of the DNA, that the two strands being complementary, if separated by heating, come together when appropriate conditions are provided. So on heating we can denature the DNA, separate the DNA strands, but under suitable conditions again they will join. In RNA at second position OH group is present, which makes it more changeable and easily degradable. RNA is also known to be catalytic in some reaction. It speeds up the rate of the reaction. Therefore, DNA chemically is less reactive and structurally more stable. As compared to RNA, DNA is less reactive and more stable. So, among the two, DNA is the better genetic material. The presence of thymine at the place of uracil in RNA. In RNA, in place of thymine, uracil is present. Because of that, it gives less stability to RNA. Both DNA and RNA are able to mutate. They can show mutations. They can evolve also. But RNA being unstable, it mutates faster. So viruses have RNA genome. They have RNA as a genetic material, and having shorter lifespan, they mutate faster and evolve faster. RNA can directly code for the synthesis of proteins, because first DNA forms RNA and RNA forms proteins. But DNA is dependent on RNA for the synthesis of proteins. The protein synthesizing machinery has evolved around RNA. So from the above discussion, we can say that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better, because from RNA directly proteins are synthesized. For stability, DNA is there. For transmission of genetic information, RNA is better. With this, I think this is clear. We will continue in the next class. Thank you.